All right, here we are, video number 2. Now we're actually going to start to talk about those individual steps. Now, you guys have seen some of these steps before. As a matter of fact, you probably know prophase, metaphase, anaphase, and telophase. But those only make up a tiny portion of what cells do. The vast majority of their life cycle, which we call a cell cycle, is what we know as interphase. Interphase, the resting or the growth phases between each cell division event. Now, just to say that it's resting doesn't mean that this isn't a closely regulated process. We're going to be very careful about choosing exactly what stage of the cell cycle that a cell is in, including very carefully controlling whether or not a cell chooses to divide. It's when cells divide out of control and unregulated, and they continue to divide, and you get this extra mass of cells in a particular area, which forms a lump, which you might even call a tumor, like in cancer. Well, we want to talk about some of the ways that this is regulated, but we want to talk also about exactly what happens in each phase. So there are two major groups. We have interphase, which is composed of gap and synthesis phases. And then we have mitotic phase, which is composed of the phases of mitosis and cytokinesis. So let's talk about interphase. About 90% of the cell's actual life cycle can be divided between G1, S, and G2. So G1 stands for the first gap phase. It's after the cell has just divided. And remember, every cell comes from a cell before it. So any cell that's entering into G1 has just divided. And it's going to need to go through a large growth phase. It's going to synthesize a bunch of proteins. It's like a new little teeny tiny baby cell, and it needs to grow up and mature and learn how to do all those adult cell things. Many cells at the end of the G1 are going to be asked, would you like to go into G0, which is kind of like a long-term stationary type phase where you just keep doing what you're doing, or do you want to divide again? And much like how you guys probably aren't ready to replicate yet, well, most cells aren't ready to replicate either. And so the majority of cells are going to go into G0. They're going to wait until a signal tells them to divide. If they receive a signal that says, I want you to proceed to the DNA synthesis phase, or S phase, this is when the genome and the centrosome actually duplicate. I want to say that again. The genome is duplicated and centrosomes are duplicated in the synthesis, or S phase, of interphase, not in mitosis. In the G2 phase, the cell is going to undergo additional kinds of preparations, knowing that it's committed to going through mitosis. These are things like dividing the mitochondria and other cell prep for those actual mitotic events. So during which phase of cell division will the cell double in size? Tell me in Socrative, and pause here if you need some time to think about it. This would be G1. And during which phase of cell division will the cell produce maximum numbers of transport vesicles that fuse with the plasma membrane? If you're not sure, draw a picture. Why would a cell produce transport vesicles that fuse with the plasma membrane? Well, transport vesicles are composed of two things. They're going to have the components on the inside, like, for example, a glycoprotein that was made by a ribosome on the rough endoplasmic reticulum. But they're also going to be bound by a membrane. As a cell goes through the process of growing, not only does it need to make more cytoplasm and more DNA, but the membrane on the outside also needs to grow. So when vesicles from the inside of the cell merge with the plasma membrane, they are also going to mix their lipids in with that plasma membrane. So as the cell is growing in G1, there are going to be other processes that are feeding this, making more lipids, distributing them, making more proteins, distributing them. So all these things you've learned about, all these structures in the cell, all the ATP that needs to be used, because those motor proteins are burning ATP in order to move the transport vesicles along microtubules. You hadn't even forgotten about that yet. All right, there are lots of connections. You still need to be able to connect organelles together, and I am going to ask you to do that on the final exam. As we're going through these videos today, I want you to try and think about some of the things you already know that are connected to this here. All right. So let's go back to the things from today's material. During which phase of cell division will the chromosomes and centromeres replicate? Well, since the S phase stands for synthesis, hopefully you told me about the DNA synthesis step of interphase. Mitosis is the stage where those duplicates divide. So I want you to think about mitosis as division. Nothing gets made in mitosis. We just divide the stuff we already made. All right, here are stages of mitosis. Prophase, prometaphase, which you may not have learned about in high school, but is an important one that we're going to consider here. Metaphase, anaphase, and telophase. So if you take those first letters, P, P, mat. PP-mat. This is how you can remember the order of these and the fact that you need to include a prometaphase between pro and metaphase. By the way, there is a fantastic walkthrough of these images in your textbook. If you don't have your textbook open, you should go get it, and you should open it up to page 186 if you're using InFocus, or page 238 if you're using Sunflower. G2 is the last phase of interphase. So we're talking now not about the cell growing. That's when all those transport vesicles and proteins and everything's being made and walked through the cell and all that good stuff. And we're not talking about synthesis here. We're actually going to look at DNA synthesis in a later step. We're going to draw out replication forks and polymerases. It's really fun. We're talking about what has happened after you've already made everything you need to make. How do you prepare? Well, the cell's already doubled in size, the chromosomes have already duplicated, the centrosomes have already duplicated, so the cell needs to divide. We have not condensed our DNA, so the DNA is still uncondensed, and you'll notice that the blue in this microscopic image, which is another form of fluorescent light microscopy, the DNA is still filling the entire nucleus. You'll also notice that even though the centrosomes have duplicated, you can't actually see each individual centrosome yet. It's not going to be until prophase when those centrosomes begin to spread apart. And if you take a look here, you'll see that there are actually two individual little centrosomes that are beginning to appear. You also might notice that the DNA, which was once evenly distributed throughout the nucleus, is starting to kind of make these little points, and that's because these chromosomes are condensing down into chromatin. And that traditional X form that you are used to looking for is actually starting to appear. These centrosomes are going to begin to spread apart, and you're going to be able to start to see these individual chromosomes. That is prophase. Now, prometaphase is going to be an important intermediate. And if you know what happens in metaphase, then you should already have an idea of what's going to be coming. By the time you get to prometaphase, there's clear distinction here between these centrosomes. But you're also going to have the nuclear lamina, those intermediate filaments, begin to break down the nuclear envelope. They'll actually regulate this breakdown. And during this phase, that nuclear structure you're used to seeing is gone. The microtubule spindle can actually reach into and touch the DNA now. So in prometaphase, this microtubule spindle was still outside. So even though it's beginning to separate and look more like a spindle, this is still in the cytoplasm and the DNA is still in the nucleus. But in prometaphase, these spindle fibers can actually begin to connect to the kinetochore proteins that are on the centromeres of these duplicated chromosomes, sister chromatids. And you can see that animated nicely down here, kinetochores that are being attached to these individual centromeres. Now, each one of these is going to be attached. So each one of these pairs of sister chromatids is going to be attached to both spindles, one chromatid to each. So take a look at this pair of sister chromatids here. These kinetochores at the centromere, one of them is attached to the top spindle and the other is attached to the bottom. I want to make note that this organism here would be an organism that has 3 chromosomes and is diploid with 2 copies of each. The big chromosome here includes one chromosome number 16 from mom and one chromosome number 16 from dad. That's why you get a copy of blue eyes from your mom and a copy of brown eyes from your dad. Right now, this isn't a big deal for you, but it's going to be when we start to contrast mitosis and meiosis, so remember this slide for later. In metaphase, all of those sister chromatids are going to line up at what's known as a metaphase plate. The metaphase plate is not a true thing. It's not a real line. It's not an actual physical plate that is in the cell. But what happens is there's an important checkpoint. And that important checkpoint says that every single sister chromatid pair is correctly attached to each of both spindles. If you don't actually pull them back and forth and line them up in the middle like this, then we know that something's wrong. So microtubule spindles are actually pretty dynamic, and the kinetochores, which are the proteins attaching the chromatids to these spindles too. So what happens during prometaphase and into metaphase is that we actually pull and push. We pull and push and pull and push and pull and push and pull and push, and eventually line all those chromatids in the middle. But let's say, for example, this spindle was gone, one of my spindles, and I only had a single spindle that was attached to the chromosome. When I went through the push and pull and push and pull and push and pull, I would only ever pull in one direction. And so that misaligned chromosome would be a signal to the cell, not everybody's in the middle, because not everybody is correctly attached to the spindles. And that means that you're not going to correctly divide your genetic material into daughter cells. So once we have that correct signal, once everybody is actually in the middle, those cohesins, the proteins holding the sister chromatids together, very quickly separate. They get cleaved, and the 2 sister chromatids flow apart to the opposite poles of the spindle, and now you have a cell that through telophase and cytokinesis can divide its genetic material and its cytoplasm into 2 new cells. Telophase is when the daughter cell's nuclei begins to form. So our nuclear lamina gets involved again, and we start to actually recreate that process, that new nucleus. Those spindle microtubules also depolymerize. Microtubules are polymers of tubulin, and so they're going to start to shed those little individual tubulin components, and that's depolymerization. Cytokinesis involves actin filaments. Those actin filaments, which we recall are on the outside of the cell in that cortex arrangement, that outside can actually begin to pinch together, and that's actually going to result in a pinching of the cytoplasm, which separates it into the 2 distinct daughter cells. All right. So because we lined up our 2 versions, mom's chromosome and dad's chromosome, the same chromosome, in this case our example is number 16. So chromosome number 16 duplicated, both mom's duplicated and dad's duplicated, and then split. So each daughter cell is going to keep a copy of mom's number 16 and a copy of dad's number 16. If any of those cells were to divide again, you would duplicate mom's number 16 chromosome and you'd duplicate dad's number 16 chromosome. Both would line up at metaphase plate and separate. This is going to be different from when we do meiosis. All right, I want to show you a video so that you can see this process in action.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid.
Note this pushing and pulling. The fact that the chromatids are moving in both directions is because their kinetochores are attached to a spindle at both sides. You're also going to see that there are two types of microtubules in the spindle. There are kinetochore microtubules, which are responsible for this tugging and pulling, but there are also spindle fibers that push the poles apart later on. And you'll see this if you watch for them.
As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome.
Cohesins
They are
are
moved toward opposite
dissolving.
poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart.
Those are those spindle fibers, those spindle microtubules.
The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
All right, so we're going to finish up just by doing a couple of Socrative questions in this video. So let's start with this one. What is the purpose of mitotic cell division? So pause here, read all the answers, and feel free to skip ahead and do the next couple, and then I'll go through and explain them. Remember, I don't care whether or not you're getting them right. That's not part of your grade. but I do want to see if you're understanding what's in the video. So please pause here and try doing them without needing to use your notes. All right, welcome back. Hopefully you did the next couple of questions, and let's talk about them. So the duplication of genetic content, that's actually going to occur in the synthesis phase of interphase. So that's not a purpose of mitotic division. The organization of sister chromatids on microtubules is, and that's part of what happens in pro-metaphase. The separation of sister chromatids equally into two daughter cells, that's what happens in anaphase. The division of homologous chromosomes to cut the number in half in each daughter cell is meiosis. So that's what we're going to be talking about on Saturday or on Thursday. Note this new term, homologous chromosomes. That's different. We're going to learn about another type. And the synthesis of macromolecules and cell growth. That's what happens in these gap phases. All right, so we're going to stop and you guys are going to go ahead and continue on with this by looking at 10.3. If you've already done your Socrative, that's great. If not, please take a break, come back.